All right, so this upcoming week in lab, you're going to be doing column chromatography, and so I'm going to go into some of the differences in different types of, or in the different common biological column chromatography columns. So, for starters, chromatography is a separation technique based on different interactions of a compound with two phases, a mobile phase and a stationary phase. Now, as the components follow through a supporting medium. The components of this, the mobile phase, the solvent or a solvent that flows through a supporting medium. That mobile phase is generally going to be your protein extract, a protein sample that's been processed in a particular way. So it's basically just where your protein, when you're doing protein or column chromatography for proteins, what that's going to be solubilized in. Your stationary phase is going to be a layer or coating of the supporting medium that interacts with the analytes. Now, the supporting medium and the stationary phase, those two are kind of combined into one thing. If you notice, the components are a mobile phase versus stationary phase slash support and supporting medium. The stationary phase is the part that actually interacts with your proteins, whereas the supporting medium is kind of the vehicle that holds that stationary phase in place. Now, whenever I think of the supporting medium, I always think of small either crystals or small insoluble beads of a sugar. Now, on that bead, you're going to find a molecule that interacts with the mobile phase or interacts with your protein. Now, protein purification, well, first and foremost, the protein must actually be harvested. So the entire cell is harvested if the protein is an intracellular protein. And what you need to do there is you need to lyse the cells, which we've done. We use that sodium phosphate buffer. We blended up our beef heart tissue, and then we went through our centrifugation steps. That releases the entire contents of the cell whenever we did that. Now, alternatively, there are some proteins that are extracellular, meaning that they are translated and then they are basically secreted from the cell. The culture medium is collected if the protein is an extracellular protein. So when you collect that, you don't have to worry about the cellular material at all. Instead, you just harvest basically the growth medium. Now, that just depends on what that protein is, and that's or what your protein of interest is. I would say more times than not, you don't study extracellular proteins, not by choice or anything like that. I mean, if you could have a choice, it'd be far easier to study an extracellular protein, but not, or lots of proteins are simply maintained within the cell. Now, separating the components within that extract. So similarities between proteins allow the separation of proteins from non-protein material. Well, one way that can be done is through protein precipitation. There are salts that cause proteins to settle out of solution, which, if you think about what we've done in our purification technique, we've used ammonium sulfate to precipitate proteins. Another way that you can separate or fractionate proteins is using filtration. So filtration, it's kind of like using a coffee filter, but with very, very small pores. So microfiltration or ultrafiltration or membrane filtration are all ways that you can separate molecules based on their size. If they make it through the filter, great. If they do not make it through the filter, then you kind of divided and conquered based on size of molecules there. And then last but not least, you can separate molecules based or through centrifugation. So how does something precipitate into that pellet or not? Another technique that's used commonly is diafiltration and dialysis. Now, what these rely on is equilibrium. Dialysis is a technique that basically use these very fine, it looks like a snake skin. You use this tubing that has very, very small pores in it. You put your protein extract into there. And let me go ahead and draw kind of a dialysis tubing bag. So if you look off to the right here, I've got a dialysis tubing or a dialysis baggie right here. And that's a bag it looks like either a produce bag from the grocery store or a snake skin, and that it's very the material clings together. Now, what you're going to do is you're going to put a clamp on one end of it to seal it closed, and then you're going to put your protein mixture into that and then clamp it on the other end to seal it closed on both ends. So you're going to have a protein sample within this dialysis baggie, and you're going to place that dialysis baggie into a larger container. Now, what this is related to is it's all reliant on equilibrium and concentration of different substances. You're going to have a high concentration of protein within this tubing, a low concentration of protein outside of this tubing. And what's slowly but surely going to happen is protein or some substance is going to leave the dialysis tubing and populate this larger container. And so slowly but surely, it will elute or dilute from that dialysis tubing into that larger container. And so ultimately, the concentration of those two substances will equilibrate. And what's really helpful for this is this allows you to kind of clean up a sample. You get rid of them, either get rid of a metabolite or get rid of some contaminant in your sample. So dialysis is another way of separating substances and kind of cleaning them up in some respect. Now, chromatography is what we're really focused on because that's what we're going to be doing. It allows for the sorting of proteins based on size or how they cling to or dissolve in various substances. Now, types of chromatography. We have size exclusion chromatography, also known as gel filtration chromatography. Now, what this uses is it uses beads with very, very small pores in them. So I always kind of draw, here's my bead, and then here's like a little tunnel or channel. Now, a very small protein, what it's going to do is it's going to enter that pore, and it's basically going to kind of be in contact with the entire interior surface of that pore and migrate through that bead. A large protein, on the other hand, well, it's going to see that opening where that channel is, and it's going to kind of like if you imagine if you were shooting a basketball, regulation size basketball on a kid size basketball hoop. Well, you'd shoot that regulation size basketball in that kid's hoop, and it would hit the rim and not go anywhere. It wouldn't migrate through. So it would rim out. So it would hit and then go to the side. So large proteins do not migrate through gel filtration chromatography columns very slowly, because they don't even enter the beads. What that means is they hit the bead and move forward. They move down that column. So gel filtration chromatography shows that your largest proteins migrate through the column at the fastest rate. Your smallest proteins migrate at the slowest rate. Now, here are a couple of different materials, and here's a cryo-EM image of a gel filtration bead on the left-hand side of the screen. Now, that's kind of the material that your proteins are migrating through. That kind of structure there is made up of one of three different materials. It's dextran, a homopolysaccharide of glucose residues used for small peptide separation. Polyacrylamide. These gels are prepared by cross-linking acrylamide, and so you're going to be able to separate slightly larger molecules with those. Your beads can also be made up of agarose, which are linear polymers of D-galactose and 3,6-anhydro-1-galactose. So the value of these, and what I want you to remember, is dextran is very effective for separating very small proteins, whereas agarose is effective for separating large molecules from one another. Now, if you look at this slide at the bottom here, I've highlighted the dextran columns. The Cefidex term, or the Cefidex name, that's kind of not an industry standard, but it's a brand name effectively. These are going to be effective for separating molecules that range from 0.05 kilodaltons all the way down to 0.7 kilodaltons. So that's the same as 50 daltons all the way up to 700 daltons, 700 daltons. Now, to put that into context, an average amino acid, so a singular amino acid has a molecular weight measured in daltons of about 110 daltons. So we're talking about something that's smaller than half of the size of an amino acid, all the way up to 7 amino acids. So dextran is really effective for separating molecules that are that small. It can also be helpful for separating molecules up to 600 kilodaltons, but that's kind of your functional range. Agarose, on the other hand, agarose down at the bottom, they're effective for separating molecules that range from 10 to 4,000 kilodaltons, 70 to 40,000 kilodaltons. And so you're talking about something that's, if it's 10 kilodaltons, you're talking about a polypeptide that's about 100 amino acids, all the way up to 40,000 kilodaltons, which would be, oh boy, math escapes me, 4,000? Yeah, 4,000 amino acids. I think that's right. Anyway, so there's your separation difference there. Again, in all of these cases, regardless of the material, the larger molecules are going to elute from that column first. The smaller molecules are going to elute from that column last. So what does gel filtration look like in chromatography, or in a chromatography column displayed at the top? And then what's visual, or the visualization of that, is what's known as a chromatogram. If you look on the left-hand side at the top, what we have is we have our sample, a mixture of proteins loaded into this column. There's low and high molecular weight proteins. What's going to happen is they're going to be migrating through this column. Your large molecular weight proteins are going to migrate through the column, and they're going to elute first, which is displayed in this third visual here, these peach balls there. Whereas our smaller proteins are still making their way through. Now, that conceptually is displayed in this chromatogram down at the bottom here. Now, anytime we look at a graph, or we always want to orient ourselves to what are the axes. Our y-axis here is a optical density or absorbance at UV 280 nanometers. So that's telling us if there's protein in the sample, because UV 280 reads, or absorbance at 280 nanometers gives us information about tyrosine and tryptophan. If we see those amino acids, that's a good indication that we do indeed have protein there. Now, our x-axis, on the other hand, what this is showing is kind of the time progression. If we look at it at our origin, we have 0 minutes all the way up to infinity minutes. So the very first minute where a sample was collected, the second minute, third minute, fourth minute, we're progressing from left to right there. So what this shows is our high molecular weight proteins are going to elute earliest. Our mid-range and our lower molecular weight proteins are going to elute as time goes on. Now, the important part about gel filtration chromatography is you only care about the time. You don't have to add anything special to get this protein to elute from the column. It's just a matter of waiting, giving those proteins time to interact with the beads and basically adhere temporarily, or for a longer period of time, or very, very transiently. Our largest proteins they spend the least time in the column, they elute first. Our smallest proteins spend the most time interacting with the beads, and they're going to spend the most time in the column. So another chromatography technique similar to gel filtration chromatography, but a little bit different, is ion exchange chromatography. In ion exchange chromatography, you've got two different major types. You've got cation exchange chromatography and anion exchange chromatography. Now, what makes these different is we're not dependent on, we don't care about the size of the proteins. Instead, what we care about is the charge. So what you see here, I'm going to draw your attention to the upper right-hand corner. We see a white bead. This is our positively charged ion exchange bead. So that has a positive charge, therefore it will attract negatively charged proteins. This is an example of anion exchange chromatography. Anion exchange has positively charged beads. Okay, now I'd like to go to the upper left-hand corner. We've got our mixture of proteins, and that mixture of proteins is going to migrate through our column. And if we've got this same positively charged bead, what we're going to have happen is we're going to have some proteins that elute from that column almost immediately because they have no charge-charge interaction upon which to basically hang around. And that's displayed by protein X, these asterisk-shaped proteins, displayed right here. Our y-axis is a measure of our UV280. Our x-axis is time, but then there's one other element that we have to consider here, and that is this other pink line, which is displayed as salt concentration. What that is it is showing our sodium chloride concentration increasing. Now it's increasing gradually. Over time, our sodium chloride concentration begins to increase. And what happens and what you observe there is that other proteins, as is displayed with this this small red ball here, and then this larger peach ball here, they're going to elute at different times, but they're eluting because increased concentrations of salt. So what that means is that this protein right here that I've just circled, protein Y, elutes with, let's say, 0.5 molar NaCl. And then let's say that this point right here is where our salt concentration is 2 molar. Okay, so protein Y elutes with 0.5 molar NaCl. And then protein Z elutes with 1.5 molar NaCl. What we see is that, or what we can take away from this, is that protein Y can elute with 0.5 molar. It can also, therefore, elute with 1.5 molar NaCl. Because if all it takes is 0.5 molar NaCl, then 1.5 molar fits the billing as well. Now, protein Z, on the other hand, that does not elute with 0.5 molar NaCl. Instead, it requires 1.5 molar NaCl in order to elute from this column. So the importance of this is it gives you a good way of separating proteins from one another. If you were trying to purify protein Z and you had a mixture of protein X, Y, and Z, you would be able to say, well, I'm going to get rid of all of the proteins that are not mine and the ones that I'm not interested, X and Y, by washing my column with 0.5 molar NaCl. I could use 1.0 molar NaCl. I could get rid of those proteins, and my protein Z, would stick on that column because it's going to stay on that column until my salt concentration is 1.5 molar. This is known as a gradient elution profile. So we are gradually, there is a gradient of salt concentration. We are gradually increasing our salt concentration to elute our proteins. Now, a couple of examples and things to consider whenever you're doing ion exchange chromatography is the pH range and the type of chromatography column it is. What is it going to be? Is it going to be a cation exchanger? And what is the pH range that it's going to have? If you take that methyl sulfonate there, the second one to the bottom, this is a strong cation exchanger. So it's going to maintain a negative charge so long as the pH is between 2 and 12. If you go above 12 or below 2, it's not going to be effective as a strong cation exchanger. Now, affinity chromatography relies on the ability of proteins to bind specifically and reversibly to uniquely shaped compounds known as ligands. This is the sort of chromatography technique that is very effective because you can kind of tailor make a chromatography column for your interest. So if you're trying to purify a protein that specifically interacts with amino or alpha aminohexyl AMP agarose, you can take this molecule, this whole thing right here, and permanently affix it to the beads. So to your supporting medium. So what that means is that you are going to be very selective. You are going to be able to get proteins that only bind to that specific molecule. If something doesn't bind to that, flows straight through. Now, the affinity chromatography column that we're going to be using is this one right here. Cipricon blue. This is a very effective molecule for binding a very wide range of proteins, including those that bind NADP or NAD plus, which is our lactate dehydrogenase. So in addition to ion exchange, gel filtration, and affinity chromatography, there's also hydrophobic interaction chromatography, which is basically operating on the idea of how hydrophobic regions of a protein are going to interact with other hydrophobic regions. I would say that in protein chromatography, this is a little bit less common than the other three, but still this has value because you do work with hydrophobic molecules. You do work with molecules that have limited solubility in water. And so it is worthwhile and valuable to have this type of separation technique. Now, what that's going to look like is not too radically different from what we've seen with our other chromatography techniques. You have these beads that have like an alkyl hydrophobic ligand. A hydrophobic region of a protein is going to bind to that and adhere to that. And what's going to happen is you're going to have to elute these by using a high salt buffer to disrupt those hydrophobic interactions. So that's actually going to be where we go ahead and stop our different analyses of our chromatography techniques. With that said, make sure that you watch the other video, or if you haven't already, make sure that you watch the other video where we talk about the worksheet a little bit. All right, well, have a good one.